All right, welcome to this second video on horizontal gene transfer. In this one, we're going to start by looking at the process of bacterial transformation. Now, this is something that you've done in a lab before in your intro biology labs here at Regis. If you recall giving bacteria the P glow plasmid, that made them glow a nice, lovely green color when you lit them up using a UV light. So that was the process of transformation. In contrast to conjugation, transformation does not require direct contact. And it could best be described as the uptake of free DNA from the environment by bacterial cells. I'm going to draw some bacteria down here, a few bacteria floating around in their environment. And as bacteria die, their cell walls break down and their DNA can be released into just the extracellular space, right? So you have these bits of broken down DNA, some plasmids floating around in the environment. Now, if our bacterial cells are what we call competent cells, that is, if they're able to take up free DNA, then these competent cells will do so, and now we'll have an extra piece of DNA in our bacterial cell. Often this DNA will just get kicked out because it doesn't have anything useful, but sometimes what can happen is that DNA can get integrated into the bacterial chromosome. So if we have our bacterial chromosome here in green, this piece of DNA can get put into our bacterial chromosome and generate what we refer to as a transformed cell. So our transformed cell has taken up that DNA from the environment that's right here in blue and has incorporated it into its own green genome. Now, not all cells are able to do this. In fact, only a small subset can do this in nature. And it's one of the ways that a number of bacteria have acquired genes to produce a capsule. But we do this all the time, and we can do this quite easily in the lab. We can chemically treat cells to make them competent, and then add plasmids to the media our bacteria are growing in and induce them to take up the plasmid and start producing whatever proteins are coded for in that plasmid, just like P-glow. Well, transformation was the simplest of all three horizontal gene transfer mechanisms. Transduction may be the most complicated. Transduction does require direct contact, but in this case, it's contact between a bacteria and a bacteriophage. And what's going to happen is that bacteriophage, when it infects a cell, it's going to pass on some cellular DNA to its next bacterial host. This process requires a bit more sophisticated drawing than I can do, so I'm going to use some slides to demonstrate this process. Okay, here at the top in our first step, we have our bacteriophage and it's infecting our bacterial cell. And we already know how replication of bacteriophages work. So it's injecting its genome into our cell. And as this cell is infected, its DNA begins to be chopped up. So we can already see here in our second step, this bacterial genome has already been fragmented. And as our little bitty purple phage genomes replicate, the kind of teal-colored bacterial chromosomes get broken into smaller and smaller pieces. When it's time now to assemble our new bacteriophages, most of them work properly and they package up phage DNA into those bacteriophage capsids. Sometimes, though, the process goes wrong and we have some bacteriophages that have actually put a part of that cellular chromosome into the phage capsid instead. What's essentially happened is a mistake in packaging or a mistake in the assembly process, where cellular DNA gets packaged into viral capsids instead of phage DNA. And this becomes important when the next round of phage infection occurs. When our newly released bacteriophage, let's say this one right here, that has some cellular DNA packaged instead of phage DNA, attaches to a new cell, It can still go through those first few steps where it injects the DNA into the new host cell. But because this is not phage DNA, we're not going to get any new bacteriophage made. Instead, what can happen is that this piece of cellular DNA can get integrated into the chromosome of the new host cell. So it can recombine with the cell's chromosome, giving that cell an extra bit of DNA. And as this cell, or a transformed cell, or a cell that went through conjugation divides, it passes this new DNA on to all of its daughter cells, so that every subsequent cell arising from this one has that same piece of DNA. So what kind of genes get transferred with transformation and transduction? Well, transformation in nature is almost entirely the acquisition of genes for capsule. There's not a whole lot of other instances of transformation in nature. Again, in lab, unlimited uses. We can make bacteria take up genes for whatever we're interested in. And over here's our pea glow plasmid that hopefully you all remember from intro biology. Transduction we do see more often in nature, and it has some pretty big implications for human health. A lot of toxins are acquired by transduction, like cholera toxins come from a bacteriophage. A really good example that we can actually see is diphtheria toxin. Diphtheria is a disease you will probably never have because you've been vaccinated for it as a part of your diphtheria, tetanus, and pertussis vaccine regimen. But this toxin kills the cells in the back of the throat here. And as those cells die and immune cells come in to try to clear up the infection, it forms this nasty sort of pussy looking pocket at the back of the throat. That gross stuff is actually called a pseudomembrane. And if it gets large enough, it can block the entire back of the throat and make it so that our patient actually suffocates. Pretty nasty. Other examples of transduction happening in nature include the passing of genes for sugar fermentation, which is great because it allows bacteria to live in a wider variety of environments and with a wider variety of sugar substrates, and those for antibiotic drug resistance. Okay, let's do our rapid review. Let's start with the beginning. Conjugation requires direct contact between two bacterial cells. Uses a sexpilus to transfer plasmids or HFR transfer. Next up, transformation. No direct contact is required, and it's the uptake of DNA from the environment. And last, but certainly not least, we have transduction. This requires direct contact between a bacteria and a bacteriophage, and it passes cellular DNA from one cell to another. So there you have it, the basics of the 3 ways that bacteria can acquire new genes that are from a non-parental source.